I would now like to invite Dr. Dhanya Lakshmi, ma'am, PhD in Medical Biology at Walter and Eliza Hall Research Institute, Melbourne, Australia, who will be discussing with us about the functional test for autoinflammatory diseases.
I'd like to thank the organizers for giving me the opportunity to talk here on functional tests for autoinflammatory diseases. And I'll start with a disclaimer. I'm no expert in this field. I'm just like taking baby steps in understanding how to do functional validation for genetic variants in autoinflammatory diseases. So we all know when we do next generation sequencing, we get a lot of variants, and then we use all computer tools to actually filter out them. And at the end, we end up having certain variants which would be classified as rare variants, and these variants are classified into pathogenic and likely pathogenic and VOUS, variants of uncertain significance, and benign and likely benign variants. So I think I discussed these aspects about the variants yesterday. So anyway, we are interested in these variants of uncertain significance. So to prove whether these variants are actually disease causing, that's where we actually need the functional genomics. So why do we need to care as clinicians? So why do we need to care about functional studies? Because of this particular fact that we are now doing these genetic tests rampantly, and we are ending up finding a lot of variants. And what we actually anticipate, or what we really want, is this is not working. Yeah, this scenario where we actually want to give a definitive diagnosis to the patient and the identified variant matches with the clinical phenotype. But sadly, that's not what happens most of the times. Most of the times we end up having a novel variant either without a matching clinical phenotype or with a matching clinical phenotype, but it's still a novel variant. Or we might end up finding a variant in a gene, in a new gene where disease association has not been previously identified. So that's when these functional studies come into play. So that's the relevance, or that's why we should care about functional studies as clinicians. Yeah, sorry for this messy slide. This is my attempt to actually simplify stuff, but since I am no expert, I was not able to anyway simplify it. So before planning functional studies, we should actually we should always remember about the inheritance pattern of the condition. So inheritance pattern is something like our shadow, like it never leaves us. So we have to always remember to approach a clinical condition by understanding the inheritance pattern, and we should try to look at what happens at the genetic level, whether the variant is causing a loss of function, or whether the variant is a dominant negative autosomal dominant variant, or whether it causes a gain of function. We should also understand what happens at the protein level, and we should understand so here you can see that even if the function, if at the genetic level, if the variant is a loss of function variant and it results in an absent protein, it can either lead to a deficiency of a particular enzyme in a particular pathway, but it can also sometimes end up in hyperactivation of another pathway. So why we need to know all this? Because based on this information, we can plan the functional validation for genetic variants. Now, functional studies for autoinflammatory diseases, we could do it in different ways. Like there is no like a single straightforward answer. And we could model these diseases like using cell-based model systems, or we could use mice models or different models which are suitable for modeling that disease. We could do transcriptomic analysis to study the mRNA, and we could do flow cytometry analysis. We could do inflammasome activation to if we think that the variant is affecting the inflammasomes. We could do cytokine profiling and enzyme assay in certain specific conditions like DADA2, and we could also do, look at the protein by using Western blot. So this is just like a basic plan or basic plan that you can have when you think about functional validation for autoinflammatory diseases. So Now, the next part of my talk would be to discuss my limited experience in doing or trying to characterize some of the genetic variants in autoinflammatory diseases. So, we had this project which was funded by the DBT Wellcome Trust India Alliance, where we tried to evaluate, we collaborated with clinicians all over India, some of them are here in this meeting as well, and we tried to do exome sequencing for around 196 families, and we could achieve a definitive diagnosis, by which I mean pathogenic or likely pathogenic variant in known disease causing genes in around 58 families, which amounts to roughly 30%, which is, I think, the diagnostic rate of exome sequencing across different cohorts. So, and we also identified 3 probable candidate variants, which means these variants were never previously reported in human diseases and in genes causing human diseases. So, so what happened is that, so now then we also ended up finding variants in certain genes which were classified as variants of uncertain significance. So this is when I specifically thought that I need to learn more to characterize these particular variants, so that we can actually develop a capability or develop our capacity to solve this huge issue for these variants of uncertain significance. And that's what actually took me to Australia to do a PhD in functional characterization of variants. So I'm currently actually working on one family from this particular cohort. And so there was this family. So I'll discuss discuss our approach in 2 families. So this was the first patient with recurrent systemic inflammation. This patient was referred to us by Dr. Suma. So he had recurrent arthritis, abdominal pain and rash since 5 years of age, and he was seen at 11 years of age, and he was born to consanguineous parents. And we did, because our approach was an exome first approach for the project, we did exome sequencing, and we did not identify any variant in known disease causing genes, but we identified a homozygous variant in the PMVK gene. And here you can see that the proband is homozygous and the parents, both the parents are heterozygous for the same variant. And the next step that we did is we went and searched literature, and we tried to find out what this PMVK gene stands for. So PMVK encodes for the PMVK enzyme, which is an enzyme in the mevalonate kinase pathway. So we already know about mevalonate kinase associated, MVK associated diseases, which is an autoinflammatory disease associated with this pathway. And at that time, when we did the literature search, we found that there was a case report which was published where they just claimed that it was a new autoinflammatory syndrome, and not much functional validation was done in this particular study at that time. So we thought we will do the functional validation for the variant that we identified. And so, so the same technique. So we understood that the inheritance pattern was autosomal recessive. We thought that this missense variant in this homozygous missense variant in that particular gene is probably causing a loss of function of that variant because it is an enzyme. So we thought that the protein level, the protein might be actually absent, or it could be having a reduced activity. And then we knew that it could be resulting in a deficiency of this enzyme, but it leads to hyperactivation of pyrin, because protein isoprenylation is actually important to keep pyrin inflammasome in check. So that's how we decided to plan our functional experiments. But by the time we planned our experiment, somebody else already published a paper where they identified a girl from Turkey who had similar features with systemic inflammation and who had a homozygous variant in PMVK gene. So here what they did, so they wanted to check they checked for the enzyme activity, because our hypothesis is that this is a loss of function variant. This could be a loss of function variant resulting in decreased enzyme activity. So they have shown that the PMVK enzyme is actually absent in the patient when compared to healthy controls. And they also did a Western blot to show the same. The PMVK protein was absent. And they also did an assay which is known as a prenylation assay, where they looked at unprenylated proteins. So here, I mean, there is actually not much of a difference between this healthy control and the patient, but this was what they did. And what we did was, we tried to look at, we collected the peripheral blood mononuclear cells from our patient, and then we looked at the IL-1 beta levels by ELISA, because here what happens is, like if protein isoprenylation is not there, then what happens is pyrin will get activated and IL-1 beta will be released. So we just checked whether the IL-1 beta levels in the unstimulated peripheral blood mononuclear cells shows an elevated level in the proband. We stimulated it with LPS, which is a TLR4 agonist, and again we see an elevated level in the proband in comparison with the unaffected parents and the healthy sibling. And we know that simvastatin is an inhibitor of HMG-CR. So we added simvastatin. So when you again inhibit this pathway, we are actually naturally expecting an increase in IL-1 beta levels, which we can see here. And GGPP is a byproduct of this particular pathway, so we added that. And then we see that there is a decrease in IL-1 beta secretion, which clearly indicates that this is the pathway that was involved for this particular patient. So that is one story, and now we have the next story. So this is the other patient who was again referred by Dr. Summa. So this is the patient that I am actually currently working on. So he had recurrent systemic inflammation and myositis, and he had a homozygous variant in this gene, HMG-CS1. This is a rare variant which is absent from population databases. And this particular variation, that is the substitution of arginine at 89th position with tryptophan, actually it results in a loss of interaction with glutamine 121 and asparagine 137. And our hypothesis was that this variation is actually in the dimerization surface. This HMG-CS1 exists as a dimer, so this variation is actually in the dimerization surface. So our hypothesis was this affects the stability of the protein and it results in decreased enzyme activity. So we already know that HMG-CS1 is the first enzyme in this particular pathway, and we already know about the various disorders that have been associated with this particular pathway. So we have PMVK and the MVK, which are already known autoinflammatory conditions associated with this pathway. So next was our effort in identifying other individuals with additional individuals with variants in HMG-CS1, and we used Gene Matcher, which is a platform which all of you can use, and you can put up your variants, and then we got a match we got actually 2 hits. So one was from a researcher in Australia who had 5 individuals from 4 unrelated families. All of them had homozygous or compound heterozygous variants in HMG-CS1 and rigid spine. But the other family was from Sri Lanka, and this was from the NIH, and they were 2 brothers who had the same homozygous variant as the Indian patient that we have, and they also have like myositis and systemic inflammation. So here this family looks like having a different phenotype in comparison to the family that we have. But we already know, like in this pathway, mevalonate kinase deficiency is associated with a neurological phenotype, and it is also associated with an inflammatory phenotype. So it could be like the disease could be driven by HMG-CS1. So what we already knew was the same information. So we thought that this particular variant is in the dimerization surface. In contrast to the variants described in patients with rigid spine, they were actually away from the dimerization surface. So our hypothesis was this variant in the dimerization surface is affecting the activity of HMG-CS1, and as I said earlier, when HMG-CS1 is deficient, it will result in a decreased amount of geranyl pyrophosphate, which will actually decrease the prenylation of Rho A, which actually keeps pyrin in check. So there will be activation of pyrin, which results in elevated level of IL-1 beta and IL-18. So to test our hypothesis, we actually developed a knockout model, a cell-based knockout model for HMGCS1, and we treated them with all these TLR agonists, and we measured IL-1 beta and IL-18 as the end products. So here you can see that the HMGCS1 knockout cells show an elevated level of IL-1 beta and IL-18. So this just means that when HMGCS1 is deficient or absent, it is actually resulting in inflammasome activation. Now the next hypothesis was, we thought that it is pyrin dependent. So what we did is we knocked out pyrin in addition to HMGCS1 in the THP1 cells. So here you can see that when we knocked out both pyrin and HMGCS1, the IL-1 beta elevation came down, which indicated indirectly that this IL-1 beta elevation is mediated by pyrin. And we also did prenylation assay with the help of one of our collaborators, Mike Rogers in Sydney. And this is a pretty cool assay that he has developed. And this is to check the levels of unprenylated protein. So here we can see that the knockout cells, our HMGCS1 knockout cells, actually show a defect in prenylation. So this clearly shows us that this particular gene is involved, in that particular pathway, results in a prenylation defect, and the inflammation there is probably driven by pyrin. So we are actually working, our work is ongoing, and that's why I'm not sharing that part of the data, because we are doing work on that. And so that was about two different situations in which we planned these functional assays. Now, those were for individual families, but you could actually plan functional assay for conditions like CAPS. For example, this slide is not required because you already know about it. So there is a postdoc in our lab, Dr. Shoya. So she went and searched in InFEVUS and ClinVar for variants in NLRP3, and she found that like majority of the variants are of unknown significance. Only like very few percentage of variants are actually classified like this. So NLRP3 is an inflammasome which requires the presence of an ASC. And when ASC actually assembles, it forms a speck in the cell, and this can be assessed by an ASC spec assay, and which Shoya did for all these variants. So what she did is, she made these variants and she transfected the HEC293T cells with those variants, and they treated them with nigericin and treated them in, cultured them in a lower temperature. Then she used MCC 950, which is an NLRP3 inhibitor, did FACS to check for the ASC specs, and then she has, this data is going to be published soon, but what she did is really cool, I would say, because now with her functional assay, she has reclassified many of these VOUS into likely pathogenic. So these variants were actually originally classified in InFEVUS database as VOUS, but with the spec assay, they were reclassified as likely pathogenic, and you have the same phenomenon in case of variants from ClinVar. So I just wanted to stress on this, that this sort of functional validation studies would help in actually improving the databases, and soon this data would be available for use for clinicians. So you could actually check your NLRP3 variant, the ones that you get as VOUS, and then you could check with the variant list, which will be soon updated into a database. And so I feel that functional testing is actually like a bridge between bench and bedside, and it's essential for precise diagnosis as well as for personalized medicine in case of autoinflammatory diseases. With that, I'd like to thank my lab mates. I can't stop here without acknowledging them, because as a clinician, my learning curve to work in the lab was is so huge, and without their support, I would not have been able to do any of this work. So So a lot of gratitude to my supervisor, all my lab mates, my PhD committee, then DBT Wellcome Trust India Alliance, which gave money to study those families, and my mentors, Amita ma'am, because she was the one who helped me with the project. She was the mentor for this project, and my previous supervisors, Dr. Girisha and Dr. Anju, and our collaborators, Dr. Suma, Mike, and Vaishnavi, who actually worked with me on the project back in Manipal. Yeah, thank you. Thank you.